And the timer isn't working, so I'll just go ahead. So again, thanks for the opportunity to talk. I really no conflicts regarding to this talk. So I'm basically talking about this paper that came out earlier this year. The first head and neck cancer to make the cover of Nature, along with 305 co-authors, including Jenny Grandis and Adele El Nagar, who are my co-chairs on this project. There are many other papers in this area, but in the 12 minutes I have, I'm going to just talk mostly about the Nature manuscript. So the hypothesis behind the TCGA head and neck and all of its tumors was to detect all of the somatic variants in cancer that occur at the 5% level. To do this, we needed a population of 500 patients, which we achieved. Actually, we have a few more than 500. And we did whole exome sequencing on all patients, whole genome sequencing on 50 patients, low-pass whole genome sequencing on 100 patients, RNA-seq, and those are the parameters for depth of sequencing. Additional platforms included microRNA by sequencing, protein by RPPA, copy number by the AFI-SNP chips, and DNA methylation primarily by the Infinium. I believe it's the Infinium array. In terms of the product, a product of this endeavor, all of the TCGA papers have shown a plot like this, which is the list of significantly mutated genes. And I'm just free associating up here anyway, so I'll just run down the list a little bit. So what did we learn about head and neck cancer? Well, one thing we learned was that CDKN2A or P16 is mutated in roughly 20% of cases, but I'll tell you for a fact that CDKN2A is lost in almost every single squamous cell carcinoma. There may be just a couple of exceptions, and those include HPV positive tumors. The other so what this says is in a canonical, really driver tumor suppressor gene, mutation is only one means of loss. I showed you this morning loss through methylation of the promoter of the exon 1 alpha, also loss through homozygous deletion, and there are obviously other methods for loss, but just to keep in mind that the actual rate of loss of tumor suppressor genes is underestimated just by the somatic mutations. FAT1, this is a gene probably no one in here has heard much about, but this is arguably the second most commonly mutated gene in head and neck cancer and linked to several pathways, including beta-catenin, and very interesting target. P53, one thing TCGA showed is that P53 mutation rates are actually much higher, and I think we showed this also for lung squamous cell carcinoma. Probably, and then remember there's some HPV positive tumors in here, so almost every tumor is also has a loss of P53, except for maybe 15 to 20% of patients. I'll come back to that later. Caspase-8, this is a really interesting target, and it lost in a coordinated way. I'll show that later. Ajuba, another gene that had never been reported to be mutated in cancer, a very interesting pathway. I won't talk about that. PIK3CA, obviously, I'll get back to this a couple of times. Notch1, which has been elegantly reported by groups from MD Anderson, Pittsburgh, and the Broad several years ago. KMT2D, which is for some reason a rename of MLL2. I don't know why it got renamed, but that's MLL2. NSD1, another fascinating gene. This is a fascinating gene, and as far as I can tell, it's really only lost in squamous cell carcinoma of the head and neck. It's methyltransferase, and loss is associated with profound alterations in DNA methylation, and there's one slide on that. I don't think I'll have much time to talk about it. Well, this is an interesting gene. So HLA-A, we also reported mutations in HLA-A and HLA-B in lung squamous cell carcinoma, as well as beta-2 microglobulin. Now, the one thing I really have to underline about this, and if I have time, I'll get back to this as well, is that HLA is a really hard gene to call mutations in. I mean, obviously, there's a lot of variability in the population. I think you have to look at these mutations with a high degree of skepticism. It's hard to call mutations. But it may be real, and it doesn't show up in all tumors, because if it was really, just a false positive, it would have shown up all the time. It doesn't. It really has just shown up in lung squamous and head and neck, but still have some concerns. TGF-beta2, which is really great, because this is the one gene that I think from the one credible mouse model. HRAS, this was known, but there's some new stuff that came out of TCGA, FBXW7. Won't say much more about that. RB1, one of the interesting things about RB1, I mean, RB1's well-known tumor suppressor gene, but what's interesting is that when it's lost in head and neck cancer, most of the patients that lost it had HPV infection, which is sort of interesting, in the setting of E6 and E7, where we already have a sort of a driver, tendency to lose RB. through E7, they feel more pressure to lose RB, which is, I mean, it's just interesting. And I don't think you would have necessarily predicted that. PIK3R1, which we don't talk about that much, but just points out that there's a lot more going on in PIK3CA just than PIK3CA mutation. There are different kinds of PIK3CA mutation. There's inactivation, or there's activation of PIK3CA. This is its repressor, and these are sort of hotspot mutations in the binding site. TRAF3, which I mentioned yesterday, also known to be mutated in other virally mediated cancers and associated with EBV positive nasopharyngeal carcinoma too. So very cool mutation. So NFE2L2, a very cool mutation I'll come back to, and its binding partner COL3, and finally a few P10s, but not making the statistically significant list. So there, there's a lot of sort of kind of a depressed sigh that there's not more oncogenes. There's a lot of druggable biology here. There's a lot of very cool stuff, a lot of integrated alterations. We sort of felt compelled to make this figure of therapeutic targets. It's in most of the TCGA papers, and you have to take it really with a grain of salt. But there's some real stuff in here too, and I will point out that the CCND1 focal high-level amplifications are not rare. They're really common. They're mostly in the HPV-negative tumors. And this may be a druggable event. I know from breast cancer, there was no association between CCND1 amplifications and response to the CDK4/6 inhibitors in the ER-positive breast cancers. Well, all ER-positive breast cancer is effectively CDK4/6 activated. So, of course, it's not a biomarker because they're all activated. This is a tumor where it's something different, and there's still some hope, although some of you may, who are further along in the CDK4/6 inhibitor story than I am, might know that I can give up that hope too, but I'm still hopeful that this may be a biomarker, and also sort of a targetable oncogene. There's a ton to say about this, but in the interest of time, I think I did mention, we threw FGFR1 focal amplifications, but I really don't think that's the case, and others before us have shown that HPV positives probably have activating mutations in PIK3CA. In about half the tumors. And the cool thing about this is that it's not all PIK3CA mutations. It's really very specifically mutations in the helicase domain. And it looks like, sort of like in smoking with KRAS and mutations associated with smoking, the base substitutions associated with tobacco, it looks like these are the ApoVec. This is an ApoVec site in PIK3CA. So just the mechanism is now coming clear. It's just a very cool story. Okay, so a couple of years ago, we also reported sort of the story for lung squamous cell carcinoma. And I think one of the unexpected findings from that project was that a finding of a subgroup of tumors that we called the classical subtype of lung squamous cell carcinoma, really almost defined by activating alterations of this gene NFE2L2, NRF2. So one of the cool things about head and neck cancer is that we find, again, looking at the sort of subtype idea and sort of borrowing some of the same lingo, especially classical and basal, those are really, and looking at correlations across the tumor types, is that we're really looking at some shared biology between head and neck cancer and lung squamous cell carcinoma that don't have time to go into in great detail, other than to say that there is this sort of pan-squamous phenotype of NRF2 activation, including head and neck and lung cancer. And what's cool is if you look, it's really entirely confined to the subtype called the classical subtype. And if you look at the classical subtype, and by the color code, I know there's a lot of going on, but the classical subtype is almost all larynx cancer. So this is a tumor that is always a smoking tumor, a heavy smoking tumor, showing a molecular alteration that's really very heavily shared with lung squamous cell carcinoma. These are the same diseases. That from a molecular standpoint, I mean, really bridging an anatomic gap that's been hard to bridge before. There's a lot in this figure. I think in the interest of time, I'll just show one other thing. Okay, so I mentioned HRAS and caspase-8. These are sort of unusual or somewhat rare events, but what's interesting about them is just a very common, co-occurrence of these two events, and often with P53 wild type, so sort of a molecular subtype coming out of sort of unexpected place, and all within this basal subtype. Just going back to NRF2, there's sort of a cool thing. There's some, a lot of evidence that NRF2 and the master oxidative stress response element and association with response to radiation and chemotherapy. So not targeted in the way that you might, hope, but again, associated with chemotherapy and radiation responses. And I'll point out, although maybe not talked about that much, but KEAP is also lost in a coordinated manner, in a subtype-specific manner in lung adenocarcinoma, and in a group that we call the proximal proliferative subtype. This is from the TCGA lung adenocarcinoma paper. And so it's something interesting about the proximal proliferative, proximal inflammatory, and the terminal respiratory units. These are the molecular subtypes that we define for lung adenocarcinoma. Well, it turns out that the proximal inflammatory and the proximal proliferative are actually equally proliferative. It's just that the proximal proliferative isn't inflammatory. And we argued for literally years about how to name all these things. So these are both proliferative, but absolutely no inflammation in this subtype. And so I'll just kind of bring this story as quickly as I can to a close. 3 smoking-related tumors, all with alterations of KEAP and NRF. So KEAP is NRF's NFE2L2's repressor, along with COL3. So these all really go very strongly together. All oxidative stress genes, with some preclinical data associated with responses to chemotherapy, differential responses to chemotherapy. And I'll just show one sort of tidbit. One of the few studies that actually has gene expression data and clinical outcomes in a clinical trial. This is the JBR10 study, which will be familiar to many of you, is the study that essentially got adjuvant chemotherapy sort of moved the ball down the field in lung cancer. Well, all of the, and these are the randomized arms, here are the 2 arms in the 3 subtypes of lung adenocarcinoma, showing essentially all of the benefit in the KEAP NRF mutant subtype, the magnoid subtype. So a very intriguing sort of clinical phenotype. Another clinical phenotype, again, showing the tumors by subtype. This is a figure that Bob Ferris really put together. And the idea originally was looking at those HLA mutations that we saw. And whenever you see a new mutation like these HLA mutations, you're sort of looking for what you expect. You look for 1 copy loss, 1 copy mutated, low expression, and you look for them to sort of cluster together by this subtype idea, because then you have some sense that the mutations are actually doing something. If they don't go with an expression subtype and they don't have that pattern, then you're sort of like, is this really real, or is this just a fake out? And it turned out that it was really hard to find much of a pattern with the HLA mutations, and it's one of the reasons why I still sort of say, I have some concerns about it. It did point out, though, that HLA, as well as most of the, many of the immune genes, are all on chromosome 6, and a single copy loss of chromosome 6 is a very common event. So I think, there's really, more to try to figure out with the genomics of the immune system. But I will point out one other thing in terms of, so just the way things are showing up here. This is the HPV status. Here's the site of the tumor, which I won't go into that too much more. Here's HLA class 1 gene expression. Here's a lymphocyte signature. This is essentially CD8 expression. And this is CD56, a marker of NK cells, and some other things. And what you'll see is, again, the KEAP NRF mutant signature with no evidence of class 1 expression, no lymphocyte infiltrates whatsoever, no CD56 positivity, and all of that as opposed to sort of the inflammatory subtype. And we've seen this through Tanguay's work and others, this idea that although PD1 and PD-L1 and some of these other markers sort of may be hard to track down, there are really very broad immunophenotypes that sort of come out of the tumors if you kind of take a step back and look. One of the cool things about this idea of tumor subtypes is maybe to go back and try to link the genomics to sort of model systems, cell of origin, stage of differentiation, and we use this model, the ALI model, air-liquid interface model, where you just take cells, let them, these are airway cells, let them grow out this early at day 3, they'll coat the plate. By day 10, they sort of start to pseudostratify, and then you get basically full pseudostratification with cilia and gland mucus formation. And if you look at the expression subtypes, you can see that the basal subtype, those cells that are present at day 3, really have very high correlation with the basal subtype, and then hence the name basal subtype. Looking at the organization by tumor subtypes as well, one thing that's also very cool is we named one of the subtypes the atypical subtype, and they're atypical because, among other things, they're the subtype of head and neck cancer that lacks chromosome 7 amplification. Chromosome 7, as you recall, and it's present in the package insert for cetuximab, don't bother staining for EGFR, all head and neck cancer is positive. Well, I think the story is actually more complicated than that. And if you look, this is a representation of chromosome 7. This is, the line here is copy neutral. And you see the tumors organized by tumor subtype, and you'll see the black line, both, this is a validation data set, lacks the amplification on 7q at EGFR. So that's the atypical subtype, whereas all the other tumor subtypes have that amplification, mostly the classical. And very interestingly, while all of the tumor subtypes have chromosome 7 amplification, only the classical subtype has this co-amplification on 7a and 7p and 7q, and this is MET, so a co-amplification of EGFR and MET. So it's sort of an interesting thing. So if you don't like gene expression subtypes, and some people don't, we can look at copy number just to sort of, again, this idea of how are the tumors organizing themselves, what is the underlying organization of these cancers, and some really cool things pop up. You just let the data organize itself and sort of clean it up using computational approaches. And this here I'm showing patients are in the columns and chromosomes are in the rows. So chromosome 1, 2, all the way down to the X chromosome. And if you let the data organize itself, you'll see that almost every sample has a 3q deletion, a 3p amplification. That's sort of a hallmark of squamous carcinoma, 8q amplifications. This is where MYC is and other things. But a couple things pop out. I'd mentioned the atypical subtype lacks EGFR amplification, and here you can sort of see that here. These are all the tumors that have chromosome 7 amplification, the atypicals without it. And then this set right here, which is very remarkable, these are the p53 wild type tumors. You see in the setting of p53 wild type, these tumors do not tolerate high levels of chromosome instability. So just another view that you can take of the genome, another way of thinking about these tumors. And I won't go into it too much, but the idea that this is among HPV negative tumors, you can really identify primarily by p53 and CCND1 groups of patients that have very poor, so these are the CCND1 amplified, p53 mutated, and the red and the black are essentially the p53 wild type. And these differences approach the difference in outcome seen with HPV positivity. I'll just skip that. I'll skip that. And so this is just sort of a summary slide. So we know about the mutations, we know about the copy number alterations, and we can sort of try using our best knowledge to put these things together into pathways. And I don't think this is the final word on this, but this is the best that we could do. Looking at the pathways that are altered in head and neck cancer, I'll just highlight a couple of the sort of maybe less appreciated ones. Certainly this differentiation pathway altered in about 2/3 of patients, mutation alterations in TP63 and beta-catenin genes, especially FAT1 and AJUBA and Notch. Oxidative stress altered in about 25% of tumors. Something going on with immunity, although I'm not quite sure that we know exactly what that is. More work to do, and certainly the RTK pathways, cell cycle, and p53 related genes. So the conclusion is TCGA, this is a big community event. I mean, 305 investigators, lots of contributors. And this is a great resource. The paper is 6 pages, the supplement is 140 pages, and then the data set is about a petabyte. So, it's awesome. And I think genomic classification can teach us a lot about how to look at these tumors. And I think I went over a little bit. I'm sorry about that. Thank you.
That was a great talk, Neil.
Thank you.
Do you have the TCGA data for head and neck? Do they have clinical follow-up so you can eventually see the prognostic implication of those mutations?
Yeah, and I think the clinical follow-up in the head and neck is pretty reasonable. They are heterogeneous groups, but yeah, I think the clinical follow-up is reasonable. Although patients are treated somewhat heterogeneously, and even though it's 500 patients, you start to slice the pie up a little thin into some of the subgroups, and there is some clinical information in there.
Because that has been a problem with the long data. that it was difficult for them to
That's right.
collect the clinical follow-up.
Yeah, a
So,
lot of the lung
if
samples.
you're asking what is the prognostic implication of mutation A, B, C, D, it is difficult for them to.
Yes, the lung data set was on the scale of pretty okay to kind of crummy. I think the lung squamous data set was on the crummier side. A lot of the tumors came from a commercial registry in Germany that really didn't provide a lot of follow-up on the clinical side. In the head and neck group, I'm involved in a number of projects where investigators call us up, and then we go back to the books and look up information and try to flesh out the data set. I think the data is pretty reasonable for head and neck clinical follow-up. But the other thing that it does is it provides a dictionary. There's only so many mutations out there, and if you look in this data set and then you look across all of TCGA, there's only a certain number of mutations. And I think it allows us, if you really care about clinical outcomes, the common variants have been described. I mean, there aren't going to be very many more. So for much less than, these cost about $10,000 a patient to do everything we have to do. I think you can now do the same thing if you're willing to, narrow down in the platforms to $1,000, maybe less than $1,000 to describe the same variants in more clinically relevant populations. And I think there is money to do that. We have a grant to work with, through the NCTN, the new cooperative group mechanism to do sequencing from clinical trials. I think there's actually plenty of sequencing out there for folks who want to do this, and we would love to see and just to give probably one of the most exciting ones are these HRAS caspase-8 mutant P53 wild type tumors are primarily they're almost exclusively in the oral cavity, and they look like they have a great outcome. So thinking about patients who you might want to back off the therapy and who are HPV negative in the oral cavity, that's a pretty easy genotype to hit, and no one has followed up on that. We think that's pretty cool.
Thank you, David.